Good morning to all. Today we'll continue with association mapping. And if you find any problem during this lecture about audio, then just put your message in the chat box, or you can WhatsApp also in our group. So yesterday we have gone through linkage disequilibrium. Association mapping is based on linkage disequilibrium. Now today we are going to see the differences between these two and details of association mapping, how it is performed. So if we compare linkage mapping and association mapping, linkage mapping is based on real-time linkage, real-time recombination. That means we cross two parents and we produce F1s. So F1 will have both the chromosome from these two parents, and we develop F2, which may be homozygous or heterozygous for locus under consideration. It relies on this real-time recombination, and you can see here blocks of linkage groups. These are the homozygous blocks from P1. These are the homozygous blocks from P2. Likewise, the real-time recombination is considered and linkage map is built up utilizing the real-time recombination. That means recombination occurring during the development of mapping population. In association mapping, if this is our gene, then we consider LD, which is based on historical recombination and evolutionary recombination. So we are considering at a time many recombination events. And you can see here, this narrow blocks as compared to these larger blocks through the chromosome, these are the very narrow blocks. And because of that, we get fine resolution. Now resolution in different mapping populations. Like if we talk about double haploid in RILs, we are talking about linkage mapping, real-time recombination. So in double haploid, as I told you, we develop, of course, double haploid from the haploid. These are the F1 individuals. Recombination occurs during F1 meiosis, and these are the this is the genetic constitution of different gametes. A particular chromosome is shown here. Different gametes are having different recombinations for that chromosome. Same thing is applicable to all remaining chromosomes. Suppose if this is the gamete of rice, only one chromosome is shown here. Other 11 chromosomes you have to imagine. Likewise, same thing is happening with each gamete. Then we double the chromosome number. So whatever recombination blocks are present in one gamete, they are in homozygous condition, as double haploids are 100% homozygous. Now, chance for recombination is given once only in F1 meiosis. And that's how, if we look at the resolution at load score of 5, that means error probability of 1 out of 100,000. With that, the resolution power is 10 centimorgan. That means you can measure the marker which is close up to 10 centimorgan up to the gene. We cannot have finer resolution than that. That means if you identify a marker in double haploids, it might be closer up to 10 centimorgan distance, closer to the gene of interest. Now, if we consider RILs, RILs are also developed from the F1, F2 individuals are raised, and from those F2s, we perform single seed descent method up to F8, F9, or F10. So many recombination events are taking place here. This is also real-time recombination occurring during the population development. So you can compare these linkage blocks as compared to double haploids. RILs have many blocks per chromosome pair, homologous chromosomes, and you can see the blocks are narrow. So it indicates more recombination. So much more resolution is possible as compared to double haploid, and the same load score of 5, we can resolve up to 5 centimorgan. That means if you identify the marker using RILs, you can identify the marker which is as close as 5 centimorgan to the gene of interest or QTLs. So that's the resolution power of double haploid and RILs utilizing linkage mapping. In association mapping, we can directly subject germplasm lines to association mapping, where we genotype the germplasm lines, we phenotype the germplasm lines for one character or many characters, and then we combine the data of LD mapping from the genotypic data and phenotypic value observed. So because we are considering evolutionary recombinations here, the blocks are narrow and we can have a resolution of 1 centimorgan. That's how we get higher resolution because we are considering many recombination events. Now the question may arise that if we do not develop mapping population, then how we can judge the distance or how we can map the genes in association mapping. So in association mapping, we do not define the individuals as parental type or recombinant type. That we only do in the case of linkage to take advantage of real-time recombination, because we are comparing the recombination profile of this individual with parent, and then linkage can be estimated. In association mapping, we are dependent upon LD. In case of LD, there are no parental and recombinant types, so mapping populations are not needed. We directly calculate the LD linkage disequilibrium between any two loci, considering two loci for allele concept. And across the germplasm line, this is the status of that chromosome across the many germplasm lines. At a time, you might be evaluating 200 or 400 germplasm lines, diverse germplasm lines. So across the germplasm lines, we focus on particular locus and we work out the linkage disequilibrium. So that's how, without mapping population, utilizing evolutionary recombinations, mapping is possible. We can directly subject germplasm panels to association mapping and we can map the genes. So here, looking to this recombination, there is a common factor for linkage and linkage disequilibrium. And that common factor is recombination. Higher the recombination, there will be no linkage. Higher the recombination, LD will also decrease. But if two loci are closely placed to each other, then there will be less recombination, there is linkage. Higher the linkage, there will be higher LD between these two loci. But in case of linkage, it is directly dependent upon recombination. In case of LD, recombination also influence closely situated loci. And there are other ways also there might be association between any two loci, even if they are situated on separate chromosomes. If these two loci are positively contributing to particular trait or fitness value of the genotype or the population, then they will be definitely associated together. They need not to be together on one chromosome closely situated. They are across the genome, they are on separate chromosomes, but they have a common trait to influence. And that's why we find association, a linkage disequilibrium between the loci which are not on the same chromosome. Now, factors affecting linkage disequilibrium and its implications. So first one is genetic diversity. Larger the genetic diversity, we get broader genetic base. We are considering multiple alleles at a time. And because we are considering many diverse germplasm lines, the LD decay would be faster. So diverse the germplasm, broader the genetic base of the population, we are calculating LD. We will find a better LD. I will find a fine resolution with that population, and it's a direct consequence of many historical recombination you are considering. So considering genetic diversity, if you are only studying 50 to 100 germplasm lines, then historical recombination event would be less as compared to studying 200 or 400 diverse collection. When you consider 400 diverse collection, definitely the number of historical recombinations you are considering are more. So we get fine resolution. So more the diversity, better is the resolution considering LD mapping. So mating system means self-pollination and cross-pollination. LD quickly declines in outcrossing species because of higher recombination in outcrossing species. Higher the heterozygosity within the population or within the genotype, there will be more recombination. And that's why in cross-pollinated species, we find narrow LD blocks. If we compare that with self-pollinated crop, because self-pollinated populations are highly homozygous, we find larger LD blocks due to low recombination rate in self-pollinated crop themselves. So nature of pollination also influences the recombination rate and indirectly it influences the LD. So in cross-pollinated crop, finer resolution will be possible as compared to self-pollinated because of the lower recombination rate of self-pollinated. So if we want to increase the recombination rate, then we have to carry out intermating in all possible combination in self-pollinated crop so that we can take advantage of fine resolution. Population structure. Population structure, when we can say anything has a structure, when it is well, when the different components of a system are arranged in different strata, that is called population structure or population stratification. So population structure, I mean the structure within the population can occur due to unequal distribution of allelic frequencies we are using to build up the population to be subjected to LD mapping or association mapping. So sometimes you collect 2 to 3 germplasms from various area and they have allelic differences. And because of that, when you mix them together for LD mapping or association mapping, because of the unequal allelic frequencies, a false LD is created. That is called population structure or population stratification. That means the whole population is divided into subclasses. These subclasses have an unequal allelic frequency. That's why they are subclasses. So we need to take into consideration this structure when we go for association mapping. There should not be population structure present. So we deal with that part. I will talk about later on how we statistically we can solve this problem. So intentionally or unintentionally, when these subgroups which are having unequal frequencies, when they are brought together, it results into population structure and it creates false LD. So it is one of the major factors in association mapping resulting into false positive. That means a marker may not be associated with the gene, but you are getting the association. That is called false positive due to population structure. Another major factor affecting the false positiveness in association mapping is genetic relationship or kinship. So false LD is created when individuals you are studying are genetically related. So for association mapping, whenever you utilize germplasm lines and whenever you build up the population for association mapping, this germplasm lines must be diverse. And for that, molecular markers can be utilized to assess the diversity. You might have 1,000 or 2,000 germplasm lines. Now you cannot subject all this to association mapping because it will increase the cost. So what we can do is we can study the molecular diversity in these 1,000 and 2,000 lines, and you can come out with the core collection. Core collection, the collection, a representation from the germplasm lines which are extremely diverse from each other. So that core collection is to be developed. You may end up with 150 or 200 or 300 germplasm lines which are actually diverse from each other. And then we subject that to association mapping. Because if they are genetically related, then it will create false LD. To understand this, I will give you my own example. You can also compare with you also. I mean, suppose if I compare the genes present in my genome with my brother's genome, and if I perform LD and association mapping, 50% of the genes will be associated. It will show LD. It's not because of the true LD due to evolutionary recombination and historical recombination. This LD is a false LD because me and my brother are sharing 50% genes. This association between the alleles is not due to the real association between the alleles, but it is due to the genetic relationship. So if individuals are related, false LD will be created. So we must select the individuals which are not related by ancestry. Genetic drift will also create false LD, and it is related with sampling error. It is very similar to the statistical concept. If there is problem in sampling, if there are sampling errors, then you will get misrepresentation about the population. So even if these two loci are in linkage equilibrium, that means there is no LD between them. If you only sample few individuals from the population, then it may create false LD. It means that from your germplasm panel, if you only choose 20 to 25 germplasm accessions for association mapping, then you may find LD in that, but it will be false LD because of the sampling error. So maximum number of germplasm lines must be utilized. Generally, it is 300 to 400 for association mapping. Migration, gene flow or admixture. Migration also increases LD. That means it creates false LD by introducing new allele. A new allele is introduced in the population, it creates false LD. So genetic drift, population bottleneck, and gene flow, they contribute to artificial LD, which negatively influences our power of association mapping. How actually migration result into false LD? Mutation also has the same effect. I don't know in detail, but migration and mutation both creates new allele in the population or introduces new allele in the population, and that's how they create false LD. Selection also creates LD. Selection, I mean, I'm talking about both natural selection and artificial selection. If two loci are positively affecting a fitness value of particular individual or the whole population, then this kind of selection will lead to permanent association between these two loci because they are positively influencing the fitness. And selection works in such a manner that if you are selecting a particular locus contributing to the fitness, the loci which are surrounding this locus, they will show association or LD with that locus which is under selection. Even if these neighboring loci are selectively neutral, even the polymorphism situated in the neighboring area of that selected locus, if these polymorphisms are neutral, they are not part of the gene, you will find LD between those. And that is advantageous because if this is so, we can utilize that polymorphism, selectively neutral polymorphism, as a marker for that particular locus. Artificial selection has also dramatic effect on LD. And it has been observed that the varieties developed so far by the breeders in a particular crop, if we perform LD mapping across these varieties developed, we find larger LD blocks in the varieties for the regions which are contributing to the yield. Because knowingly or unknowingly, breeders are selecting those genes which are contributing to the yield. So yield contributing alleles, polymorphisms will be the alleles or the yield contributing loci would be common in all these high yielding varieties. And if you perform LD, because they have been selected, you will find larger LD blocks. There are no narrow LD blocks there. So this is the effect of selection. Then recombination rate. LD is strongly influenced by recombination rate. As I told you earlier, linkage and LD both are influenced by recombination rate. Lower the recombination rate, the linkage will be present, and lower the recombination rate, LD will be there. LD is high. If recombination rate is high, then LD will decay. So there is one terminology for us, LD decay. LD decay means there might be association between any two loci at a particular point of time. But if any phenomenon, natural or artificial crossing or anything, if it increases recombination rate between these two loci, then ultimately LD will decay and these two loci will be in linkage equilibrium. So that's called LD decay. When you find LD between two loci, and when it decays because of higher recombination rate or any other factor, it decays, it means these two loci eventually will achieve linkage equilibrium. That's called LD decay. During that, if we can perform mapping, LD mapping, then we can have finer resolution. So this is also better. LD decay is good because when LD decay occurs, you can identify a closely linked loci. Because the loci which are far away from the locus under consideration, because of the LD decay and recombination, those loci will no longer show LD with your gene. So that's how recombination rate affects LD and linkage both. But recombination rate differs from one region of chromosome to another region. As previously I have given the example of centromere, and recombination rate also varies from chromosome to chromosome. All these things are to be considered for LD mapping. Mutation. It increases LD by introducing new allele. So those were the factors affecting linkage disequilibrium. Now the difference between linkage mapping and association mapping. So linkage mapping is based on family. That means we have to create biparental population. All the biparental populations are familial population developed from two parents. Association mapping can be performed in familial populations as well as unrelated populations. Only few recombination events are considered, as I told you, real-time recombination events, and here all the historical and evolutionary recombinations are considered. So many recombination events are considered. Only two alleles are studied at a time in linkage mapping because we are crossing two homozygous parents to create biparental population. So when you say two homozygous parents, considering particular locus, if these two parents are diverse, then there are two alleles present, different allele present. When you prepare mapping population, develop mapping population from these two parents, this mapping population will have two alleles maximum for a particular locus, not more than that. But in association mapping, because we are considering diverse parents or the diverse germplasm lines, there are more alleles under consideration. Here we are talking about multiple alleles. At a time, even if you're considering the germplasm lines for rice, diploid species, any diploid species, self-pollinated or cross-pollinated crop, at one locus there will be two alleles. But because we are considering the evolutionary recombinations and diverse germplasm lines, there will be more than two alleles present in your mapping the population, whether it's the germplasm panel or mapping population developed. So because we are considering few recombination events or real-time recombination events, the resolution is very low. We get loosely linked marker. And in association mapping, we get high resolution because we are considering more recombination events. Linkage mapping is, the results of the linkage mapping are limited to specific population. It is applicable to one population only. If you apply that marker trait relationship established from particular population to other population, it may not work in other population because other population, the allelic constitution might be different. So most of the time, the data obtained from the marker trait relationship obtained from linkage mapping, it is validated in germplasm lines or diverse population. But generally, we don't find this valid. I mean, most of the time, if it holds true, if you can validate marker trait relationship found out in linkage mapping population, biparental population, you can validate that in other population, it is well and good. That confirms your result. But most of the time, it will not hold true because of the allelic diversity between the populations. Association mapping, the results of association mapping are not specific to population. It is applicable to entire species because we are considering all historical recombination. We are considering multiple alleles at a time. So it is not specific to population. The results are applicable to all the species. Because we are considering few recombination events or real-time recombination, and linkage blocks are larger than LD blocks, a few markers can also do the job in case of linkage mapping. But in association mapping, because we are considering many recombination events, we require more markers. We need more marker coverage across the genome. Then and then association mapping is possible. Linkage mapping is weak design. That means it doesn't give you the accurate result as you get in association mapping. Association mapping is powerful design, takes into consideration recombination events, many recombination events. Duration is more because we need to develop mapping population. Duration is less in case of association mapping. If you are applying germplasm panels, you can directly subject them to association mapping. So the time is not required. I mean, less time is required in association mapping because mapping populations are not to be developed. Linkage mapping is very good for initial detection. With few markers, you can utilize real-time recombination and you may get loosely linked marker to the gene of interest. It's very good for initial detection, but it is very poor for fine mapping because low number of recognition events. Association mapping is very poor for initial detection because there are chances of false positives due to population structure and genetic relationships. But it is very good for fine mapping. So both have advantages and disadvantages. Association mapping is far better than linkage mapping. If association mapping conducted properly, it can give you more better result than linkage mapping. We can combine them both also. We'll see that later on. Now these are the steps of association mapping. We start with, let us say, germplasm panels which are diverse from each other. We phenotype these germplasm lines for a particular trait or many traits. Let us say at that time you might be evaluating these germplasm lines for salinity tolerance. Then we perform genotyping of these germplasm lines by utilizing any marker technique, any technique by which you can assess the polymorphism. Nowadays, genotyping by sequencing method is available. So by direct sequencing, we can genotype the individual for the SNP polymorphisms present. And once we have the genotypic data, we deal with population structure and genetic relationship and determine that by Q and K matrix or matrices to work out the possibility of false positive. We overcome that and we perform LD mapping on the basis of the genotypic data obtained from the germplasm lines, linkage disequilibrium mapping across the genome. And then we associate this LD map data with this phenotype. So we perform association mapping and we identify the polymorphisms which are associated with particular phenotype. Now, this phenotype may be qualitative trait, quantitative trait, any trait. There are certain terminologies we need to know related to association mapping and the statistics involved. So population structure, as I mentioned earlier, it is population stratification. And this stratification arises due to unequal allelic frequencies between the populations which are mixed for association mapping. There is another terminology which is called genomic control. Genomic control is utilized to assess the level of population structure present within your population under study for association mapping. So in genomic control, we utilize a set of random markers. Random markers are utilized to find out the false positive. If you utilize random markers and if you find association between marker and trait, then it indicates false association because you are utilizing random markers. You don't have, I mean, these random markers are not associated with the trait, or you don't have prior information that they are associated. You just choose random markers and perform association mapping. So you will be able to know the inflation, that is of bias caused by these kind of population structure in your analysis. You will only know the bias. You can measure that inflation. Structured association is another method by which we deal with population structure. And structured association also, it uses the set of random markers to estimate the population structure. But the difference from the genomic control is it incorporates this estimate for further analysis in case of LD mapping and association mapping. By GC, that is genomic control, you can know only the inflation caused by the structure. In SA, that is structured association, we estimate it and we incorporate that in our analysis. That's the difference. Kinship, I already told you the genetic relatedness. If there is a genetic relationship, it will create false LD because now the association will be due to genetic relationship rather than the actual real evolutionary association. Migration or admixture. So as I already mentioned, that migration or gene flow also create false LD by introducing new allele. So this Q matrix and K matrix, I will not go in detail, but these matrices are utilized to work out the population structure and genetic relationship. When we work out the K matrix, that is genetic relationship, there are two terminologies which are not mentioned here, IBD and IBS. IBD is identity by descent and IBS is identity by state. these are considered for working out the genetic relationship. Identity by descent means any two alleles present in a particular individual, the probability works out the probability that they are identical. And when these are identical, two identical alleles, they might be present by state of the genotype. That means it is this the homozygosity or identity is not due to genetic relationship. That is called identity by state. And identity by descent means these two alleles are identical because of the descent, that is genetic descent, because these individuals are derived from the parents genotypes which are related to each other. That is called identity by descent. Just for your information, these are the concept utilized to construct the K-matrix and to work out, to calculate genetic relationship between the individuals. Now, some of the statistical aspect, false discovery rate is worked out. And it is a proportion of the positive results that are actually false to the whole set of positive results statistically derived. So statistically, we can also work out the false positive by type 1 error estimation, all these things. And we work out the actual false positive. So that can be worked out by utilizing random markers. And or what you can do after performing association mapping, the markers are shuffled, and the shuffling provides the randomness. And after shuffling of the LDMAP data or the markers, even if you are finding the association, that is actual false association. So we work out the ratio of actual false positive to the statistical false positive. That gives the false discovery ratio. That also gives you idea that how much false positives can be assessed by statistical methods. How many false positive can be assessed by statistical method? How good is our method to analyze the false positive? So FDR is very important. If this can be incorporated, then we can know the false positive, because false positive is the major limitation of association mapping. And that is why association mapping is poor for initial detection, but better for time mapping. Minor allelic frequency is also important. Generally, in association mapping, alleles which are having the frequency less than 5 or less than 10%, they are not taken into account for association mapping because they cause some kind of bias to the LD mapping and association mapping. So these minor alleles are not under consideration. And that is estimated, and when minor allele frequency is estimated, and all those alleles which are having less than 5% or 10% frequency, they are eliminated from the analysis. Now, Bonferroni correction. These are the statistical solutions used in association mapping. When we go for multiple trait association analysis, that is possible only with association mapping, that you can identify the polymorphisms which are associated with many traits at a time, which is not possible in linkage mapping. Now, as I have come up with this concept. Now, in linkage mapping, multiple trait analysis is not possible because we are utilizing biparental population. And these two parents we are utilizing which are diverse for a particular trait, not necessary that they are diverse for all the traits we want to analyze. That's the problem. But in association mapping, you can utilize diverse germplasm lines, which are having the diversity for many characters. In germplasm panel, you may have 400 germplasm lines, and they might be diverse for many traits. So you can perform association mapping for multiple traits. So at that time, we are testing multiple hypotheses. With all the polymorphisms investigated, we try to associate this polymorphism with multiple traits. So there is not single hypothesis involved, there are multiple hypotheses involved. And for that, Bonferroni correction is necessary. I'm explaining all these terminologies because when you read association papers, research papers on association mapping, you will come across all these things so that you can understand why these are present in that research paper and what is the importance of those things, those statistical solutions. So we work out the error probability associated with alpha error, what we can say type 1 error. So if the same error probability is utilized, it is also error probability is also related with level of significance. If the same probability is utilized for all the hypotheses, it is statistically not correct. So that if 10 hypotheses are tested, then we divide this error probability by 10, and then we take that probability into consideration. If you understand it simply why Bonferroni correction is applied, the probability of identifying at least one significant result due to chance factor increases if you test more hypotheses at a time. If you test more hypotheses, you will get significant result, but it is due to chance factor. It is not due to real differences. So when multiple hypotheses are tested, the error, this type 1 error is divided by the number of hypotheses tested, and that is called Bonferroni correction. Because we want significant result due to real association, not due to chance association. So this correction is necessary. Now, p-value and log p-values are also mentioned in the papers of association mapping. These p-values are non-parametric estimate of false discovery ratio that can be calculated by comparing the distribution of p-values against set of markers with known influence. You know that these markers are associated with the trait under study, and some of the random markers. We also study some of the random markers, and then we work out the probability of false association, which is the ratio proportion of the significant association detected with random set. So if you are utilizing random set, that should not give you the association. If it gives you the association, that is false association. 2, the proportion of significant association detected by this known markers. So we are actually working out the ratio of the random marker association with the trait and known markers association with the trait, and that gives us idea about error probability. That gives us idea about the false discovery ratio also. And mostly this probability in logarithmic terms, they are expressed in terms of this Manhattan plot, because this looks like the buildings, multi-story building of Manhattan, which is a part of New York. It is called Manhattan plot. In this, on x-axis, we have different chromosomes. This is for rice. So there are 12 chromosomes. So if you just work out the colors present, there are 12. It indicates the first chromosome, second chromosome, up to 12 chromosome. Each dot indicates the SNP present there at particular location. And this is the negative logarithm of p-value. Now suppose error probability for particular SNP is for a particular experiment is 10 raised to minus 15 for a particular SNP. So if you work out the negative logarithm of that 10 raised to minus 15, it is so 10 raised to minus 15 means the logarithm of that would be minus 15. But we are talking about negative logarithm. So negative logarithm of 10 raised to minus 15 would be 50. Now, if you compare that with 10 raised to minus 17, that error probability is low. 10 raised to minus 17 is low as compared to 10 raised to minus 15. So it will give you the value of 17. That means the higher the value of negative logarithm of these p-values, higher the significance of that SNP. So here the threshold value of 4 is utilized, and all these SNPs which are going beyond this threshold value, they are highly significantly associated with particular trait. So that's how the results are interpreted from Manhattan plot, and that's how these p-values are utilized. Generally on x-axis we have the chromosomes, points indicate the SNPs under consideration, and on y-axis you will have negative logarithm of these p-values. Again, what is p-value? You can infer from here. It is related with false discovery ratio. Lesser the false discovery ratio, lower the p-value, it is better. Significance is higher. You are getting real association as compared to chance factor association. Now, power of association mapping and statistical analysis. So, power of association mapping will depend upon extent of LD in the genomic regions. As I told you, different regions show different recombination rate. So that is also important. That will decide the power of association mapping. Type of gene actions involved: additive, dominance, epistatic. If epistatic interactions are present, then power will go down. Size and composition of the population. If there is too much population structure present, then power will go down. If you only analyze few individuals, even if germplasm lines, if you utilize only 50 to 100, then also power will go down. To increase the power, you must utilize many germplasm lines. The population size should be higher, up to 300 to 400, and you must utilize diverse germplasm lines. Genotyping and phenotyping also influences power of association mapping. So accurate genotyping and phenotyping must be done. See, nowadays in this era of NGS and genotyping by sequencing, genotyping is not difficult. It's very easy and accurate. But phenotyping is generally not accurate. Phenotyping has never been that accurate. So one must focus on accurate phenotyping. Genotyping is nowadays very accurate and it is possible. And it is easy as compared to phenotyping, especially when you consider the complex trait like this salinity tolerance, abiotic stresses, other quantitative traits. And how you are dealing with false positive, it also decides the power of association mapping. So I'll come to that point, how we can work out this, how we can apply this. Now, there are two models used in association mapping. One is called general linear model. So this general linear model only utilizes Q, that is either population structure or genetic relationship, to avoid the false positive. That is called general linear model. Single, either of these at a times, only one, either population structure or kinship. In mixed linear model, which are much more better, they utilize both population structure as kinship to avoid the false positive statistical solution. So mixed linear model are much more better than general linear model. These are some of the softwares which are utilized for association mapping and to work out population structure. Structure and Tessel, they both are very popular softwares for association mapping. Nowadays, there are softwares for mixed models. These are the names of the softwares and website. You can visit those sites if you are interested to know. Now, these are the statistical solutions for avoiding the false positive and study different types of populations. So in this graph on x-axis, we have familial relatedness, which is increasing from this origin towards this side. The familial relationship increases. Genetic relationship is increasing. Here on y-axis, we have population structure. It is increasing in this direction. Now you might have population in your hand. Single line represent one germplasm, let us say, one germplasm. And because these lines all are merging at single, they have common center, it indicates that there is no population structure, there is no population stratification, and these lines are not interconnected, so there is no genetic relationship. So if you have germplasm panel and you are subjecting them to association mapping, and there is no population structure present and there is no genetic relationship present, you have such population, it is best population. And for that, generally genomic control is sufficient to avoid false positive. You might have population B. Here all lines are merging into one common point. This indicates that there is no population structure. But you see interconnecting, these lines are interconnected. It indicates genetic relationship. So to deal with this kind of population where genetic relationship is there, genomic control and mixed models are utilized. We have a population C where there is genetic relationship also, as these lines are connected by each other, and these are not merging at common point. There is no common center for all these lines. So it indicates population structure, and the whole population is divided into two parts because of unequal allelic frequencies. And for that, we can use structured association, genomic control, and a mixed linear model. There's another population you can see here from this graph. Genetic relationship is very low here. The lines are not connected, but population structure is very high in this particular population. You can see here because there is no common center for these lines. And for that, structure association and genomic control can be utilized. There is a fifth kind of population where there is extreme genetic relationship and extreme population stratification. There are no statistical solution for these kind of the population. And association mapping, accurate association mapping or powerful association mapping in this population is very difficult to achieve. Now, types of association mapping. There are basically two types: candidate gene association mapping and genome-wide association mapping. Simply, it is called GWAS, Genome-Wide Association Study, or GWAM, Genome-Wide Association Mapping, GWAM. So let us focus on candidate gene association mapping. This candidate mapping has some primary knowledge about gene under consideration. Through mutational analysis, through biochemical pathway analysis, or linkage analysis, we know few things about that gene. So some prior information is available. And we only focus on that limited region or candidate region to find out the polymorphisms associated with the trait. So it requires this low cost approach because we are focusing on limited region. The markers to be taken into considerations are very low in number, so that will reduce the cost. It is hypothesis driven. We know that candidate gene is present somewhere in this limited region, and it is trait specific approach. It is easier, but there is one limitation. Because you have limited your search in a particular region, you will not be focusing on other loci which might be influencing the trait. So we are missing those unknown loci. So that's candidate gene association mapping. And this is true for quantitative trait because in quantitative traits, as I mentioned earlier, the loci might be distributed across the genome. We cannot limit our search for one region in quantitative trait unless and until you have some kind of reliable primary information. It's better to go for genome-wide association mapping. So here in this case, we don't have any information about the region governing the trait, and we work out or we take into consideration large number of markers across the genome. And then we associate this LD map created from this genome-wide polymorphisms, we associate that LD map with the phenotypic trait. And genome-wide association mapping was previously not possible, but now because of availability of information about
हेलो।
हेलो।
Imanshuman. Hello.
हेलो हेलो।
ए
हाँ अभी रिकॉर्ड हुआ।
सर को बोल देना रिकॉर्डिंग सेव करने।
हाँ मैंने सर को लिंक सेंड किया शायद।
सॉरी सर।
अच्छा सर ज्वाइन कर लिया सर ने। हाँ
सर प्लीज सेव द रिकॉर्डिंग।